Gentlemen, it is indeed a great honor for me to stand here together with a man that I've been admiring for several decades. First, I need to say that this is probably a social experiment that Mike is putting us through. Now 9:30 into the night, local time here, for 4:30 into the night or early in the morning in Oslo time. And we're still here. We're experiencing great excitement. And so it was indeed exhilarating and very stimulating to listen to Erik Betzik's talk. And I'm here now to introduce to you Jon Storm-Mathisen. He's a great inventor, both in terms of inventing molecules and also in terms of inventing other people's talents and fostering other people's talents, I would say. So Jon, he's from Oslo, and he graduated from high school, or went to high school at the Oslo Cathedral High School, very prestigious in Oslo. And then he went, of course, to Oslo's most prestigious university, I would say, Oslo University, and studied medicine there. And so by the time he had finished studying medicine, he was entitled to go into military service, and he did so by joining the Defense Research Institute, where he studied neurotoxicology, among others, together with Frode Fonum. And from there, he went on to, he made his doctoral dissertation there, and he went straight on to an anatomical institute at the University of Oslo, where he has been since, and he's now an emeritus professor. So he started working with the electron microscope and invented the immunogold technique to visualize amino acids, and he's going to tell you about this himself. And so he studied molecules and like GABA and glutamate and glutamine, and together with his collaborators Ole Petter Ottersen and Nils Christian Danbolt, and later Farrokh Chaudhry, and now Linda Bergersen and a lot of others, he's been inventing one molecule after the other. I think one molecule per year, Jon.
Very exaggerated.
Well, anyway, please, I give you Jon Storm-Mathisen.
Thank you for being here at this late hour. And I start to begin by thanking those people you see here, and because they have been at it for many, many years to create things like this. And this is the climax so far. So I'm grateful for that, and I'm also grateful to all the other people who are around and have made this possible. And I want to direct this to the man here, Hall Irwin. Then on to the subject, which is how glutamate and GABA became neurotransmitters. I put the quotation mark in there, of course. So I'll talk a little bit about that. And I could mention this funding bodies down here that have supported us over the years. But first, I will send the regards of Ole Petter Ottersen, who is the rector or the president of the University of Oslo, and tell you just a few words about the University of Oslo. It was the first university in Norway, founded in 1811, just before we got independence from Denmark, and teamed up with Sweden for 100 years about. It has this budget here. There are quite a number of students, and importantly in this context is that we have 15% of the students are from abroad, and even higher percentage of the academic staff and the PhD candidates. University of Oslo is putting great emphasis on life sciences, and they are now building a large life sciences center, which will be Norway's largest and most advanced laboratory building. And they are emphasizing education, and every 3rd of September, all of the students are greeted here by the rector and the Minister of Education and the Mayor of Oslo. Yeah, so now to business. I will speak on how glutamate and GABA came to be recognized as the excitatory and the inhibitory transmitters of brain signals. And GABA's role was actually established before that of glutamate, and the recognition of glutamate was delayed for decades. There was a psychological barrier, I believe, because of glutamate's ubiquitous excitatory action, high concentration, and diverse other roles like metabolic currency, nutrient, main building block of proteins, umami, taste, and precursor of GABA. That's a bit of a list. This historic tale is largely based on my own experiences. I say that because somebody might miss some important things. But first, I will give you a few glimpses about neuroscience in Norway. And I was pleased that Mike Friedlander showed a picture of our national hero, Fridtjof Nansen, who earned his fame as a polar explorer and later a statesman and humanitarian. But here you can see him at work at the microscope in Bergen Museum, where he was a pupil of Armand Hansen, who discovered the leprosy bacillus. And he actually can be said to be the founder of the neuron doctrine, because he, in contrast to the prevailing view at the time, he said that the nervous system consists, or the central nervous system consists of separate units, namely the nerve cells, which we now call neurons. And he also understood that the activity in the nervous system was very much happening in what we now call neuropil, but which was called the dotted substance at that time. And he has a very bold statement at the end of his doctoral thesis. This is probably the principal seat of intelligence. He did not actually see the synapses because he didn't have a microscope that could have that resolution, but you can see he drew varicosities on axons. Yeah, after a lag, there was this man here, Jan Birger Jansen, who was very much inspired by Nansen. He established, together with his colleague Alf Brodal, the Oslo School of Neuroanatomy. Some foreign students have called it that. And Jan Jansen got money from Rockefeller to establish the brain laboratory, and it was modest with, if you compare with things we just heard. But this is a picture taken around 1960 when I started to work there. And Theodor Blakstad established electron microscopy, or biological electron microscopy, in Norway, and he got money from the NIH to buy the first electron microscope, which you see here. And then we see Per Andersen, who founded Norwegian neurophysiology, and we'll see some of its famous students afterwards. Kirsten Olsen and Per are the only ones of those who are still alive. Fred Balberg was he was what was referred to as Director DI Grazie of the Anatomical Institute. And here we have some of those people who were there in a little genealogy. But this man here, I think, is quite important because he was behind hippocampal physiology and the hippocampal slice, which has been so much used to study physiology of nerve cells generally, and it was in his lab that the LTP was discovered, and he also did a lot of work on that. Here you see him, how he looked when I came to know him. And here we have some of his PhD students, and two of them became Nobel laureates. Here we have the postdocs, or the doctoral students at the time, who were the first authors on the several papers on the grid cells. And I took these pictures when they got the Kavli Institute in Trondheim. And here you can see Edvard and May-Britt Moser, and you can also see Menno Witter, who was a professor of anatomy in Amsterdam, but he moved to Trondheim to work with those people. And that was long before they got the prize. And here is Terje Lømo, who actually made the first observations on LTP. Here is my mentor, father of hippocampus anatomy, I call him. One of his colleagues, Gary van Heusen, wrote this at the time when we had a little jubilee. A wonderful individual, the gene for all modern investigations on the hippocampal formation. Theodor Blakstad worked out the pathways that are now so well known. And when I started to work with him, he had me to stain the hippocampus with acetylcholinesterase staining. And this, at that time, this was the only method that was available to show nerve cells and axons on the basis of the transmitter that they contained. And it was not until Falk and Hillarp, I think the very first publications came out in 1959, where they could show the catecholamines directly in the microscope. But that was not possible that these two substances could be the main transmitters of inhibition and excitation in the brain. So what could they be? Actually, these people who I mentioned demonstrated how the inhibitory synapses in the brain look and where they are located. And Per Andersen was at this time at Eccles laboratory in Canberra, and they published 3 papers in Nature, similar to, or at about the same time. And here you can see these inhibitory synapses, which are known as forming synapses of gray types 2 on the cell bodies mainly. Yeah, so then what was the inhibitory transmitter? Eugene Roberts in 1949 discovered GABA. There was this amino acid that was special and occurred at very high concentration in brains. And it was thought to have metabolic roles, and it even was thought to be a metabolic wastebasket. Later, it was found that the GABA shunt, through GABA goes a bypass of the Krebs cycle, which accounts for about 10% of the flux, total flux of the Krebs cycle. And in hindsight, we can think that this is because GABAergic inhibition is so prevalent, so abundant, that it explains that 10% of the flux of the Krebs cycle goes through it. But at the same time, or slightly later, Ernst Florey and collaborators in Canada discovered also a principle that was in the brain, which was called factor 1. And it was uniquely produced in the brain, and it inhibited the stretch receptor of and also mammalian neurons. And it was identified after a while as identical to GABA. So then we had an inhibitory principle And this could mean that GABA actually could be the inhibitory transmitter in the brain. But was GABA selectively produced in inhibitory nerve endings in the brain? And that is something I had the pleasure of being involved in showing together with Frode Fonnem and Fred Wahlberg. Fred Wahlberg had shown that he had shown the projection of the Purkinje cells, which was known then to be an inhibitory pathway. And we measured the formation of GABA by the enzyme glutamate carboxylase, which you can measure in this way here by dissecting very small samples and putting them into half a microliter or so of incubation fluid containing radio-labeled glutamate, and then the enzyme clips off the CO2, which you then hunt over, to drive it over to the other side here by having the sulfuric acid drop falling down and collect it here, and you can measure the activity. So this was before there was any immunocytochemistry and these things that we use now. So here is this paper that I'm a bit proud of. This was performed in 1969, but was published in 1970. And here you can see the projection of the Purkinje cells, and they end, some of the terminals end in the dorsal part of the lateral vestibular nucleus. So and we dissected then bits from there and bits from the ventral part, which does not have such terminals, and we measured the GABA production. And it turned out that the GABA production was much higher dorsally, and when we made lesions to destroy these axons, then we lost the activity in the dorsal part, but not in the ventral part. So this was the first demonstration that production of GABA was connected with inhibitory neurons. So when Eugene Roberts was having a symposium to put together a book on GABA, he asked me by cable if I would like to come. And that was very unusual question, so I said yes, of course. And I presented there, and one thing that was, one concept that was much talked about there was the concept of disinhibition, because you can have two GABA neurons in a row, and then, of course, you can switch off inhibition, and then you produce excitation, which is what you do with the Purkinje cells. You can regulate the activity, and for example, the inhibitory GABAergic neurons in the cerebral cortex can turn down the Purkinje cells so that the output comes from the cerebellum, gives reduced inhibition. And Eugene Roberts was also an amateur artist, and I happen to have this picture on the wall, which I think is a little bit connected to this inhibition, and at least this cover, I think, hints to that, or to the effect of inhibition. Roberts' group isolated GAD after a while, and they made antibodies to it, so then you got a tool to demonstrate GABA production or the enzyme in situ, and it turned out that there are at least two different forms. Those are the most well-known, and the one with the slightly higher molecular weight is distributed throughout the neuron, whereas the GAD65 is concentrated in the nerve endings, and it is also more regulated. So that is probably a mechanism by which GABA is adjusted. So here you see the Purkinje cells, and they have this pinceau, which they call it in French. It's a cluster of nerve endings that sit exactly at the axon hillock, where the action potential gets generated. So they sit exactly where they can best control the output. And then a little bit later, we made an immunohistochemical method for amino acids and showed that GABA can also be used as a marker in the microscope, both the light microscope and the electron microscope level. And here you see a pyramidal cell in the hippocampal region, which does not contain GABA, but the terminals on the surface do. And here is the cell body of one of those inhibitory neurons in the hippocampus. This could be a so-called basket cell, and if you look carefully, you can see there are some blebs there, which are the inhibitory GABA-containing terminals, which you also find on the surface of those cells. And then here is the electron microscopic picture, where we can see that some of the terminals, not all, but some of the terminals have the reaction product. They contain GABA. This is a little bit ugly, but it is because we have to smash the tissue with Triton in order to get the antibodies into the synaptic vesicles, but we believe this is what it means. Then identification of GABA receptors was dependent on finding good blockers and agonists. And some of the more important are bicuculin, which was found by Graham Johnson, and muscimol, I think mainly identified by Krogsgaard-Larsen, and they collaborated, the Australians and the Scandinavians collaborated. And muscimol is from Amanita muscaria. That's such a beautiful mushroom. The fly agaric. And then there was baclofen, which was synthesized by Ciba-Geigy, chemist, in 1962. And I think it was in order to have a substance like GABA that was lipid soluble so that it would gain access to the brain. But it turned out that it does not work on the same receptors as bicuculin and muscimol. The one that performed a lot of work on this was Norman Bowery. So some people said that it's GABA B receptors. That means not baclofen, but Bowery. But I think it's because they were B after A, because the ionotropic GABA receptors are called GABA A, and the metabotropic G protein receptors are GABA B receptors. But Eugene Roberts said at that meeting, which I refer to, you cannot run a nervous system with only inhibition. And everybody thought this must be glutamate, but nobody had the correct or had the salient evidence for that. In the meantime, Andersen, Blakstad, and Lømo had actually made physiological and electron microscopic investigation which showed that the excitatory nerve terminals and synapses were on thorns, on spines, and they were invariably of the gray type 1, which has this which are characterized by the postsynaptic density, PSD, as it is named now. Yes, and this seemed to be the case for wherever they looked, and they gave examples from 4 different pathways in the hippocampus. And now, glutamate was actually identified in 1866 by, and it was isolated from wheat gluten, hence the name. And Ikeda crystallized it from broth. And he found that it had a very strong taste, and he thought that was a good taste. So he called it umami, which in Japanese, I'm told, means pleasant, savory taste. Then the next things that we can mention is that Hans Krebs of the Krebs cycle did a lot of studies on glutamate in the brain, and he found that glutamate was in brain at high concentration, and also that brain slices accumulated glutamate to a large, very avidly from the medium. And then Hayashi, the Japanese, injected glutamate into the brain and also into carotid arteries and observed that it created conversions. So he thought, suggested on the basis of that, that might be an excitatory transmitter in the brain. Jeff Watkins is a very important guy for the next development. He was a chemist, and he got the idea when he was in Boston at Harvard, actually, that he should try to figure out about the chemistry of the brain, because there must be something important to be found out there. And one physiologist he turned into said that he'd better go and study with John Eccles in Canberra. And he was himself a native Australian, so he went there. And he teamed up with David Curtis. John Eccles was just converted from his very stubborn view that synaptic transmission in the brain was electrical rather than chemical, but he had realized that this was not the case. It must be chemical, so he was very pleased that there was a good chemist who came along to work with him. And Curtis and Watkins started to work on this, and they found that glutamate was indeed excitatory on the cellular level, and they wrote up a paper which they sent to Nature. At that time, that was surface mail, which means by ship from Australia to London. And it took about 2 months. And in the meantime, they did more experiments. They put glutamate onto the Renshaw neuron, which inhibits the motor neurons. And it was known at that time that the Renshaw neurons receive cholinergic input from collateral of the motor axon. But glutamate excited those too. So they thought that must be entirely unspecific. And wherever they looked, it always excited. So they wrote a revised version of the manuscript and said that, yeah, it's exciting, but it's probably an autotransmitter. And they sent it by ear to Nature, and it was accepted and published. Watkins, no, that was not that one. It was Curtis, Phyllis, and Watkins. I came out in 1959, and after that, Curtis went around on meetings. I've been there with him many times, and he always said that you must caution, but there are so many unspecific things about glutamate. So it lasted quite a while, but in the meantime, Watkins went on and he tried to find drugs that could block the actions of glutamate, and he found N-methyl D-aspartate. But in the meantime, some other Australians, Graham Johnston and colleagues, and also Snyder Group in the US, observed that you have glutamate uptake in brain slices, and they thought this was due to nerve endings because you can observe it in synaptosome fractions. So that was something, and it also came around that this uptake system did not discriminate between L-glutamate, L-aspartate, and D-aspartate. And D-aspartate was good because L-glutamate, when you incubate tissue with that, it gets changed to lots of different things, but D-aspartate is hardly metabolite. So that became the favorite labeling, with radio-labeled D-aspartate became a favorite thing. And it was actually then used by us and others as a marker of what we thought might be glutamatergic nerve endings, either in slices or in crude synaptosome fractions. And we worked on hippocampal connections, which Blakstad had worked out. And he showed that neurons in this region here, they project their axons to where you see the stippling, and there are some other neurons that project over here, that both on the ipsilateral and the contralateral side. And he also showed that you have a so-called perforant path that comes from the neighboring area entorinalis, where we know now that the grid cells are, and they project. There are two parts of it, and they project to the outer and the middle part of the molecular layer in the dentate area. So we worked on those. We looked at those projections, and here is the aspartate uptake, and followed by autoradiography. So you see that the target area of the excitatory neurons, excitatory fibers from CA3 neurons, they end here, and they have aspartate uptake. And if you make a lesion to destroy those neurons and remove this thing here, then this uptake disappears. And also, if you make a lesion of the perforant path, then you can see that this uptake here also disappears. No, not that this uptake that you have here, which is less strong than that over here, but it gets even less strong when you lesion the perforant path. So much for that. So that indicated that these pathways that were then thought to be, that were known to be excitatory, they were associated with this high affinity glutamate and the aspartate uptake. And at about this time, most people thought that glutamate was actually a transmitter, and there was a meeting that I helped to organize with Graham Johnston and Peter Roberts in Jerusalem, and it was in 1979, and it came out a little bit later. And in that paper, in that book, Per Andersen had a paper, and we can hear the master David Curtis behind there, because you can realize that from the way he phrases himself. It is therefore possible that glutamate acts as an excitatory transmitter for several of the afferent systems impinging upon CA1 pyramidal cells. Yes. I heard somebody say at the meeting that John Eccles showed that glutamate was the transmitter in brain. So I looked up to papers by John Eccles and glutamate, and I found this one. That was the only one I found in PubMed. But it was very gratifying to see that he actually, first he used this scheme which he has from Per Andersen, which shows all those fiber pathways which I mentioned, and then he says liberation of the synaptic impulses in the perforating pathway cause the liberation of the synaptic transmitter glutamate. And it says 56, that is a paper of mine. He also refers to another one, which is by Neidler and Kotman, which showed they did something that was very challenging. They dissected these target areas of from fresh slices, and they incubated them with the polarizing solution, and they collected the amino acids that came out, and they showed that it was glutamate and aspartate that was released calcium dependently, depending on the existence of these pathways. So that was quite interesting at the time. But then the question was glutamate selectively concentrated in excitatory nerve endings in the brain? And notably, would it be concentrated in the synaptic vesicles? Could we show that? And we addressed that by making antibodies to various amino acids, including glutamate. And this is not so strange that it was possible to do it, because when you fix tissue with aldehydes, glutamate and other amino acids gets tethered to tissue macromolecules, so they sit there. And of course the products are something that haven't been in the body before, and the immune system recognized them as foreign, so we got nice antibodies, and they turned out to be specific to the individual amino acids, which we could show here by treating amino acids with glutaraldehyde together with beads that had brain protein adsorbed on them. And then we could see that antibody to glutamate reacted with glutamate, but not with the others. Slight cross-reaction with the glutamine, but this is something you could absorb away, and also GABA has specificity. Here we can see some of this pattern that you recognize from the autoradiography of the aspartate that I showed you previously. And here, for example, the mossy fiber endings are rich in glutamate. And if you look at them with the electron microscope, you see the same thing. They are bags full of small blobs. It's ugly for the same reason that I explained with GABA, but I think this is the first demonstration that glutamate is enriched in synaptic vesicles in nerve endings in an excitatory pathway. And at the same time, the Japanese Naito and Ueda published that synaptic vesicles isolated from brain have a ATP-dependent uptake system which accumulates glutamate. Later, we worked more on this, and Vidar Gundersen, who you see here, in particular, used the immunogold method to localize amino acids in a rather better way. Immunogold doesn't have it has a resolution that is of the same order as the size of synaptic vesicle, but you can do various tricks to show that if you record what sort of structure that is under the center of this immunogold particle, you can find that both glutamate and aspartate, representing gold particles, are much more concentrated over vesicles than over the intervening cytosol, whereas the opposite is true of glutamine, as expected. And this study by Gundersen also revealed other things, namely that the amino acid is exocytosed, and you can actually drive it out entirely, empty the nerve endings with prolonged depolarization. And this is exocytotic mechanism because it is blocked by tetanus toxin and also by removing calcium and adding magnesium. And also, if you supply glutamine during the depolarization, you get a picture like this. That meaning that glutamine can sustain the level of glutamate during the polarization. So, and then also, aspartate behaved in the same way, which was a little bit intriguing, and indeed, aspartate and glutamate are in the same nerve endings, because here you have two serial sections that are treated with the two different antibodies, and you see they're both labeled. So the meaning of this is still enigmatic, because it turns out that aspartate is a weaker agonist for the receptors and only for the NMDA receptors, whereas glutamate works on all the different glutamate receptors with good reason or by definition. So it's not, this is a puzzle that has not been resolved. I think perhaps aspartate could have a modulating action, perhaps affecting the speed of desensitization or something like that, but that remains to be investigated. Ole Petter Ottersen worked with me on this, and here you can see that in a mossy fiber in the mossy fiber ending in cerebellum, that glutamate is in the mossy fiber, and glutamine is also a little bit there, but much more in the glia astrocyte. And different cell compartments are clustered in different groups when you look at the relative concentration of glutamate and glutamine. And you can actually quantify this, so that you can measure the concentration of these amino acids in the subcellular compartments. Yeah. Specific agonists and blockers are necessary to decipher the receptors, and also to show that the action, synaptic action, is due to glutamate. And this was done. Here is just a selection or some examples of the types of substances that were the most important. And I already mentioned NMDA, N-methyl-D-aspartate, and kainate, which comes from Japan, and AMPA, which has a name that nobody wants to remember how to pronounce, but AMPA is so these three have defined three groups of excitatory amino acid excitatory amino acid receptors, and they are actually of many different subgroups. And then we have the metabotropic ones, so there is a lot of different receptors for glutamate. And here I've collected some important publications on glutamate acid transmitter, but the most important, which really decided about these things, is when the receptors were cloned, and there were three groups mainly who did that: Heinemann in California, and Seeborg in Göttingen, and Nakanishi in Japan. And this happened around 1990. And then, of course, it turned out that each of the receptors have four, they are heterotetramers, and it means, and they form a channel that can be opened by binding of glutamate. And this fact that you have receptors that consist of different subunits of different kinds means that you have a lot of different possibilities of combining receptors. So it means that there is a big opportunity for neuropharmacology and for precision neuroscience. Before we knew that, we couldn't fancy that it would be a good idea to interfere with these receptors, because they were all over. They would, if you interfered, they would mock up the whole, or destroy the whole place, whole thing. But here also, this is really quite well known, but I can mention it nevertheless because it is important. You have on the spines, subsynaptically, you have mainly the two types: AMPA receptors and NMDA receptors. And NMDA receptors are not functioning when unless the membrane is slightly depolarized, because they are blocked by magnesium, and this magnesium block gets relieved when you get a small depolarization, which means that you need to activate the AMPA receptors first, and then afterwards NMDA receptors can come into function. And NMDA receptors are also permeable to calcium, so when they get into action, then you get rise in calcium. And of course, this means that you can have regulation here, and some of the regulation goes with deciding how much of the receptor is going to be intracellularly localized and how much is being localized in the membrane. And there is also regulation of the supply of the receptors after synthesis. Plasticity can also be changed by changing the size of the synapses, which we have heard about from in the talks today. And here is this old cartoon which I taken from Alenka, which shows, this has also been mentioned today, that you have LTP and LTD, and both are steered by increasing calcium concentration, but if calcium concentration rises strongly, then you get LTP, but if it rises only little, then you get the opposite effect, which is quite... oh yes, I see you have something there. Yeah. Then I think we can make some shortcuts, but I would like to show you this thing here, that actually LTP was discovered by Terje Lømo and Tim Bliss in Per Andersen's lab in Oslo in 1967, but Lømo actually had the first publication on that. This thing just mentions that the very thing that makes it possible for the synapses to be plastic, so that you can learn, also involves a danger, because the calcium rise, if it gets out of hand, it can activate all sorts of damaging things, including damage to DNA. This was the basis of the Center of Molecular Biology and Neuroscience, which Ole Petter was director for, and I was a member. Here I just will want to show you the things that I've had the fortune to be involved in that have to do with this synapse, the glutamatergic synapse, and the first one is what I showed you, that the glutamate vesicles contain glutamate vesicles in excitatory nerve endings contain glutamate, and also aspartate, and they get released. And then Niskis and Dambold isolated the first glutamate transporter and cloned it at about the same time as two others cloned two different glutamate transporters. And I think Susan Amara was also involved in that business. And then I was fortunate to have this student here who wanted to go to America to work, to do his PhD, as part of his PhD, and he teamed up with Robert Edwards. And Robert Edwards thought that it was so good that he would have more Norwegian students. But he identified then two important transporters, namely the glutamine transporter that lets glutamine out of the astrocytes, and the other class that is able to accumulate glutamine against a concentration gradient. And then I was also involved with these people in identifying VGlut2 and 3. There are 3 different vesicular glutamate transporters. And then Linda Bergersen came along and looked at the MCTs, the monocarboxylate transporters, which supply lactate to this system as a fuel, and also it turns out as a signaling substance because it has a receptor. You will hear about that tomorrow. And now I will, I think, skip very quickly to the last. Oh yeah, I did something wrong. This is what I wanted. I wanted to show you this. After all, this is supposed to be an after-dinner speech, which in Norwegian would be "Takk for maten, Tale." Thank you for the food speech, which we have at prestigious dinners. You always have somebody to do that. And what is better suited than glutamate? It's a very versatile substance. And you already heard that it has its name from gluten. And this gluten is an interesting word because it's akin to clay, and this is again akin to High German kliva, which is kli. The Norwegians know what kli is, and it means bran. And it also Latin gluten means glue. And lo and behold, Middle Greek, glia. And how could you know that the glutamate was had so much to do with glia? It is the most abundant amino acid in protein. It's also the most abundant amino acid in the cell sap of cells in general, and is an energy metabolite. It's a precursor of GABA and glutathione, which is an important substance, and it's a nutrient. And when something is important as a nutrient, it should taste good, and that's why it is called umami. And then savor. It is called savory when you translate the Japanese to English. And savor has come into English from Anglo-French savur, from Latin sapor, from sapere, to taste. And that means also to be wise. And in fact, it is in the English word sage, and in fact it is the name of our species, Homo sapiens. Isn't that good? And glutamate has functions like signaling between cells and also signaling inside cells. And that is a good end note. But at least some people think you should not have too much of it. Thank you.
Dr. Storm-Mathisen, that was a fantastic and scholarly presentation of the topic. I'm sure many of the younger students and postdocs here got an experience to learn a lot of things they hadn't seen before. For me, it was a walk through history, so much of it was fantastic. Maybe time for one brief question. I know it's getting late, but if there's a burning question for Dr. Storm-Mathisen, yes, Mike.
So when I was a graduate student, there was a professor, James Fernando, who was still studying aspartate as a neurotransmitter, and you kind of tailed off on the story there. What do you think aspartate is?
Yeah, I mentioned Naylor's work, where they did this challenging work of finding that aspartate actually comes out. And I think I said that we do not really know the role of aspartate. It's enigmatic because it is a weaker excitant than glutamate is. So why is it there? It has other functions in metabolism, but it is actually released. And the other thing that is enigmatic is that the vesicular glutamate transporters do not apparently transport aspartate. But nevertheless, aspartate gets into the vesicles, and D-aspartate also gets into the vesicles. So so how they do that is not known. But I think perhaps it has a modulating function.
Great. Well, please join me again in thanking Dr. Storm-Mathisen for a fantastic lecture.
Thank you.
That was wonderful.